Okay, so here we go with the review for the test on Tuesday. And so we'll start with, remember, I'm just going to throw in the last section in Chapter 14, 14.4. We came into the final exam before we were able to finish that, so I only had the final exam go through 14.3. All right, so the things in 14.4 is pedigree analysis. So I taught about pedigrees and how circles are female and squares are male, and shows the kids and so on, and how you trace a trait through the family, and the people that are shaded in are the people affected with that trait. All right, so that trait can be dominant, recessive, or sex-linked. And I'm just going to introduce sex-linked now, but sex-linked was actually in Chapter 14, but you actually had a pedigree analysis sheet with the 3 pedigrees on it, and you had to tell me if it was dominant, recessive, or sex-linked. I have a problem on the test like this, so you have to look at a pedigree and be able to tell based upon who's shaded in and so on. So remember, it's kind of like you make a hypothesis, then you give everybody a genotype and see if it works, and remember looking for discrepancies and whether or not there's discrepancies. So then the rest of this section was about specific disorders, so that there's recessive disorders. So recessive disorders, examples would be cystic fibrosis, sickle cell anemia, are the recessive disorders. We looked at dominant disorders like Huntington's disease, a type of dwarfism, achondroplasia. So people here that have the disorder have a big R something. Recessive, the people who have the disorder are little r, right? And then the last part of this was about genetic testing and being able to test. So we talked about amniocentesis and chronic villus sampling, where you can take a piece of the amnion. And so there we go. So review those. Okay? And that was the last section of Chapter 14. So then Chapter 15, we continued on with genetics. So Chapter 15 was about genetics. So 15.1 was the law of segregation. That's not the title. That should have gone under here. It's a law of segregation. And the law of independent assortment. So let me just review what that means. So remember, you have, this is a pair of chromosomes, and we're going to go through meiosis. Remember, the spindles attach and pull the pairs apart. So that's the law of segregation. The law of segregation say that the homologous chromosomes segregate or separate into different cells during meiosis. And so then when meiosis 2 happens, you get the sister chromatid over here, this sister chromatid over here, this one here, and this one here. So they separate or segregate. And so that's the law of segregation. The law of independent assortment is if I were to draw another chromosome pair, so let's do a green one in here. All right, so then one green would go over here, one green would go over here, and you get one green, one green, one green, one green. So we have 2 chromosome pairs. So the law of independent assortment is how the chromosomes line up. So remember, they can line up this one on the left and this one on the right, or vice versa. And so remember, that was where we get that formula 2 to the nth power, where that's how many combinations you can get based upon each chromosome pair. That's the haploid number, or each chromosome pair lining up in one of 2 different ways. And so that's your law of independent assortment. Then I started introducing to you the scientist Morgan and his flies. And so what about Morgan and his flies? So talked about how he labeled them differently, wild type versus mutant. Wild type, remember, is the most common, and a mutant is a mutation of that wild type. And he came up with that genes are on specific chromosomes. So, for example, on the X chromosome. So review that and review Morgan and so on in 15.1. So then instead of going on to 15.2, we skipped to 15.3 because it kind of flowed with this. Oops, that's supposed to be 3. 15.3 was about sex-linked traits, which kind of goes with the whole idea that genes are in specific chromosomes, an example with the sex-linked or the X chromosome. So remember for humans, so most often these are on sex chromosomes, most on the X chromosome. So examples of these in humans of a sex-linked trait would be colorblindness, certain types of muscular dystrophy, and hemophilia. And so for colorblindness, for colorblindness, these are all recessive disorders. So that means that, remember, if you're a female, you're X little r, or X little r y if you're a male. So males only have one of the colorblind genes, and because they have one X chromosome, and that's why we see these seen most often in males, all right, as a result of that. Okay, so this is what I was talking about here with the sex-linked traits, adding to the pedigree analysis up here with those three ones that three examples that I gave you in class, and being able to do that and to analyze if it's sex-linked or dominant or recessive. And then the section also talked about X inactivation, that in females, remember, one of your two X chromosomes becomes, one of the two X chromosomes becomes inactivated and becomes a bar body. So therefore, and it's random, and so you can get different cells. You get some cells showing one X, and then other cells, this X might inactivate. And so the example was with a calico cat. And that's how you get the splotches, and that's why most calico cats are females. And so that's the third section. So going back to the second section, 15.2. 15.2 was all about linked versus unlinked. Whoops, link, there's an N. Genes. So remember, linked means that they're on the same chromosome. Unlinked, we're on different chromosomes. And so if they're linked or unlinked, when you do crosses, like I had you do the P, then the F1 generation, and then I had you take the F1 and take some of those kids and do a test cross and look at the ratios in the kids. And so if they're on the same versus different chromosomes, we're going to expect different ratios in our results. And so in analyzing those ratios, we can determine if they're linked or unlinked. And so look at, I would have you look at, we did this sheet, solving linkage and crossing over problems to review about that. and we had another sheet, linked and unlinked. It was a two-sided sheet where we looked at what would happen, what would we expect if the genes were linked, on the other side if they're unlinked, and how to do that. So you'll have a problem similar to that. And so with that, going with that to look at the expected ratios, how they're different, figuring out the percent of crossing over. Remember, you take the number of recombinants that you have, the ones that wouldn't be able to occur unless crossing over occurred, divided by your total offspring, and that gives you your percent of crossing over. And how that relates then, the percent of crossing over, to linkage, making a linkage map. So I'll put using percent crossing over. So what does that mean? So remember that this is your DNA molecule. The genes are far apart on the chromosome, A and B, let's say, and this is A, and this is B, and this is, let's say the person's heterozygous A and B. The farther apart the genes are on the chromosome, the more likely crossing over is going to happen between them, or likely to occur between them. And let's say the B's switch, and the little b goes over here, and the little big B goes over there. If the gene, that A gene, was here, really close to the B gene, then crossing over would occur less often because there's less room in between there. There's less physical space, and therefore the frequency of crossing over will go down. So you can use the frequency of crossing over to figure out the order, the relative positions of the genes on the chromosome. So review that. We did an example in our notes, so look at that. All right, and so that's 15.3. So then 15.4 was all about how we get alternate chromosome numbers, the alteration of chromosome numbers. So this happens because of non-disjunction. Non-disjunction, remember, is when the chromosomes don't separate properly. So here's your replicated chromosomes. I'm going to go through meiosis. Now, normal meiosis, this one's supposed to go over here, this one's supposed to go over here. Non-disjunction occurs. Can occur in meiosis 1, where both of them go in the first cell, and neither one of them go in the second cell. So then these cells have two, let's say these are chromosome pair number 1s, they have two number 1s, and these guys here have no number 1s. And so these guys have the haploid number +1, so they have an extra chromosome. These guys have the haploid number -1. All right. The same thing can happen also in meiosis 2. So I showed you meiosis 1 occurring. I'm going to just kind of do this over here. So if it happens in meiosis 2, here's your chromosome pairs. So meiosis 1 happens properly, but now let's say meiosis 2 messes up and both the chromatids go in this cell and none go here, and this can happen properly. So these guys have the right haploid number. This has the haploid number -1. This has the haploid number +1. Sorry. All right, so that's non-disjunction in meiosis 2. So both of these create a situation where a gamete would have an extra or a missing chromosome. And so that's what we see here. So this, for instance, like if this was an egg cell and you had a sperm cell that was made properly with one chromosome number 1, the zygote, the baby here, would have 3 chromosome number 1s. All right, 2 from mom, 1 from dad. And so we can get disorders based upon chromosome number. Then you can have alteration of chromosome structure. So deletion. So this would be a part of the chromosome is deleted. So we have, I gave you pictures of these. So we have deletion where like you have segment A, B, C, and D of the chromosome, and if a segment is deleted, so you have A, B, and then D, let's say. So C was deleted. Then there was duplication. So you have missing DNA here. Here we duplicate it, so it would be like you'd have two sections of B. Inversion, where they flip and the sections flip. So like, okay, so here would be, let's say A, B, C, D. All right, so that's duplication. Inversion is a part that's flipped. So if this is your original here, you'd have A, C, B, D. So the B and the C, it's like we cut them out, took it out, and flipped it over. So that's your inversion, and then you have translocation, where it moves from to one chromosome to another, from one, they're non-homologous chromosomes. So that way, so disorders can happen because of a gene. So you inherit a gene, like, so that's how sickle cell anemia and cystic fibrosis and Huntington's, that's all how all of those are inherited. But the this is showing you that you can also have genetic disorders that are caused by abnormal numbers of chromosomes or by something wrong with the chromosomes, so the alteration of chromosome structure. So the last part in 15.4 here is we talked about disorders due to chromosome alterations. So that would be Down syndrome. It's trisomy 21, another way to say that. So it's 3 chromosome number 21s. Klinefelter syndrome. That's a male that is XXY for their 23rd pair of chromosomes. And lastly, Turner syndrome. Turner syndrome, it's a female, it's XO, they don't have a second X chromosome, so they're missing a chromosome. And then the last section of chapter 15 is about genomic imprinting. Remember, that is only an allele from one parent is expressed. So the other one is silenced. So I gave an example of the mouse gene in there. So genomic imprinting. And then the second thing with chapter 15.5 is extranuclear genes, genes outside of the nucleus. And so these are like organelle genes. So we went over genes in the mitochondria and the chloroplast. And that is chapter 15. 16 was real short. It only had 2 sections. It's all about DNA. So 16, DNA as the genetic material. So they used to think protein was the genetic material, right? And so then we did different experiments to show how DNA was ultimately proved. So that proved as the genetic material. So we talked about some experiments. Okay, so what experiments do you need to know? So we need to know Griffith, Frederick Griffith, Avery, McCarty, and McLeod, Hershey and Chase, Shargaff. Then we need to know how Watson and Crick, so we need to know what Watson and Crick did, deduced the structure of DNA and describe the evidence they used. Be able to explain the role of Wilkins and Franklin. Rosalind Franklin. And then the overall, so that's the history, and then you need to know the overall structure of DNA. Sugar, phosphate backbone, a nucleotide, phosphate group, the sugar, and the base. And in DNA, that nucleotide is made of, the sugar is deoxyribose, and the base is going to be A, T, C, C, or G. And that it's a chain of these nucleotides. So then it's going to bind to the next nucleotide and with the base, and then you have two strands of it, and it twists and into the double helix form and so on. So the overall structure of that. So then 16.2 was about DNA replication and repair. So we need to know about the semi-conservative model. With that, an experiment, we need to know Messelson and Stahl's experiment. So they put the radioactive phosphorus or radioactive nitrogen to tell whether or not the DNA of the bacteriophage was actually, the genetic material, the virus was actually DNA or RNA. Alrighty. So you should be able to describe the overall process of DNA replication, including the role of the origin of replication. I'm sorry, it's kind of sideways on here. The origin of replication and replication forks. So remember, you have many different replication sites, and that this is the start of replication. Each one of these, this is the right here, here is a replication fork. This whole thing is a replication bubble. All right, so and remember, this helps with the speed of the molecule, or of the replication, having many different origins of replication and therefore many different replication bubbles. All right, continuing on here for DNA replication, know what the role of DNA polymerases are. And how they add nucleotides. This should be DNA polymerase 3. Add nucleotides in 5' to 3' direction in the new strand. you should be able to explain the energy source. It's a C, of DNA replication. So what is that? Remember, it's the nucleoside triphosphate. Remember that you have, remember, a nucleotide is the phosphate, sugar, and the base, but a nucleoside triphosphate has 3 phosphate groups attached to it, and 2 of them come off as a pyrophosphate to provide the energy to hook the new nucleotide to the growing strand. All right, so that's your energy source. Understand and define, know what anti-parallel means. Remember, it goes in 3 to 5, 5 to 3 prime direction. And explain why continuous synthesis of both DNA strands, like both sides, is not possible. So why is that not possible when you do this? Why is that? Because remember, it always has to grow, DNA polymerase has to grow in the new strand in the 5 to 3 prime direction. And so you have, and it has to be anti-parallel, so therefore the DNA, in order to grow the new strand, the template has to be in the 3 to 5 prime direction. Be able to distinguish between the leading and lagging strand. To tell that. So remember, I had on the board something like this, and I said, okay, this is the 3 prime end and this is the 5 prime end. So you had to be able to say, oh, this is going to be the 5 prime end of the new strand, so this is going to be the leading, and the 5 prime end of this strand is going to be here, so therefore this side is going to be the lagging, and built-in Okazaki fragments. Next here. So you should be able to explain how the lagging strand is made. And the significance of the Okazaki fragments. What are they? How they're made? It has to go in the 5 to 3 prime direction. Should know some enzymes. Enzymes, so we already talked about DNA polymerase 3, adding the nucleotides. There's DNA polymerase 1, which replaces the RNA primers with DNA. So, so we have primase. We have helicase, unwinds the DNA at the beginning. We have DNA ligase, glues the Okazaki fragments together. And the single strand binding proteins. So all these guys, what's their role in there? Something I thought before the enzymes here I should have added, and we can add that here, is the role of a primer and why it's needed. So why do we need a primer? Remember, that's because DNA polymerase 3 can't add nucleotides unless there's pre-existing nucleotides. And so in order for to add nucleotides, it needs a primer at the beginning of each fragment. Okay. Okay, so then let's look at the role of telomeres. So what's the role, function of telomeres? Remember, those are at the end, and explain the significance of the enzyme telomerase. Where do we find those? Those are found in germ cells. And we also can see them in cancerous cells as well. So what's the significance there? So remember that when DNA is replicated in 3 and the 5' here, and we add the primer here to be the 5' end, and we continue in synthesis this way, and then each of these fragments have each of the Okazaki fragments here have an individual primer, that DNA polymerase 1 comes back and removes that primer, and so I'm going to show you it's now purple, so it removes that, and DNA ligase glues it all together. But what happens is when this is removed, this primer is removed, DNA polymerase can't add new nucleotides, so I can't make this purple. It can't add DNA nucleotides, and so we miss this part here, and so it's not replicated, and so that is the telomere. The telomeres at the end don't have, they don't code for making proteins or anything like that, so they're not actually genes, and so it's okay that we miss them or are missing them for a while, and so therefore our ends of our chromosomes get shorter with each cell division. And so then the last thing is that we have, just in case we have mistakes, we have repair enzymes that repair DNA that so we have mismatch repair enzymes. So if DNA polymerase accidentally adds a T instead of an A or something like that, so we have that, and so these guys proofread and repair the DNA. So you have enzymes to do that, and that is chapter 16. Chapter 17 was with protein synthesis, and so I'm not going to go over all of chapter 17. We're going to finish the last little bit of the notes of chapter 17 on Monday. Really, there's only like 15 minutes or so left, but I'll just talk about the very beginning of chapter 17 here, and then after we finish the notes, I might do a little review, pull everything together with chapter 17. So I'm not going to do it here, but I'll do the first section, the first little bit of the section here, in that you need to know the beetle, excuse me, and Tatum experiment. This is with, they use bread mold. And so basically after that, they eventually came up with the idea, they restated it, that one gene, one polypeptide hypothesis. That every gene codes for making a polypeptide. So they had the different pathways, remember, in the bread mold, where A gets converted to B, which gets converted to C and D, and a different enzyme catalyzes each of these reactions. And so each one of these enzymes then, this enzyme is made. All right, how do we make that? By gene 1, gene 2, gene 3. So there's a gene. Gene 1 makes enzyme 1, and gene 2 makes enzyme 2, and so on and so forth. And so, so they proved with this bread mold experiment that that every gene codes for one particular polypeptide. And so, remember, they changed it from one gene, one enzyme to one gene, one polypeptide, because not all proteins are actually enzymes. Okay? So then the section here goes through the difference between prokaryotic versus eukaryotic cells. So prokaryotic cells, we have two steps. We go from DNA to mRNA. That's transcription. And you go from mRNA to polypeptide. And so, and that's translation. No nucleus, remember, in prokaryotic cells, so it's all happening in the cytoplasm. All right, this is a little bit different than in eukaryotic cells. Eukaryotic cells, you have a nucleus, and you have your DNA, then you have your pre-mRNA, and then you have your shorter final mRNA strand, which then leaves the nucleus, goes to the ribosome, it's the mRNA, then we build our polypeptide chain from there. So this right here is transcription. This is RNA processing. And this is translation. So I introduced to you these processes. I introduced mRNA, and this is the section also we learned about the difference between DNA and RNA. So review that. The bases, the sugar ribose, single-stranded. This one goes out of the nucleus, DNA does not. So that's the difference between RNA and DNA, and then we also learned how to use the chart, all right, and how we read mRNA is read in codons. So each codon, remember, is a group of three. So A, U, G through three bases, C, U, 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 or U, A, U, all right, and so on and so forth. So this has three codons on there, and we read that, and that's what's used in the chart. Okay, so that was the first section there that we did. So the rest of it, transcription, RNA processing, and translation, I'll review with you again on Monday. All right, so happy studying, you guys. Have a good weekend.